Chang's lab. And he's going to be telling us about Mergeomics, multidimensional data integration.
Okay, good afternoon.
Yeah.
Yeah, team parent, yeah. So good afternoon, everyone. I'm very glad that I have the opportunity today to present you a tool that's newly developed in our lab called Mergeomics, which is able to perform multidimensional data integration to identify pathogenic perturbations to biological systems. So our presentation will be separated into two parts. The first part, I will introduce you the Mergeomics computational pipeline, and then Doug gonna introduce you the Mergeomics web server. So I'll start with my part first. So we're now in the era of data explosion where biological data kind of doubles its amount each year since 2008. More than that, we are kind of have access to a number of different publicly available databases that allows us to retrieve tons of biological data. So now the bottleneck is really how do we make sense of these huge amounts of data and how do we get useful biological insights from these data? A useful framework to apply here is systems biology framework, where we basically collect information on a systems level, which could be from the genetics, transcriptomics, proteomics, metabolites, even microbiomics. Then we could use different methods to model all these different layers of data, such like network modeling. This will help us get an idea how the variation at different layers of biology eventually contribute to the final phenotypes that we observed. So based on this concept, we developed Mergeomics. What Mergeomics do is basically it takes a series of different biological data as input, starting from some of the most commonly used association data sets, including GWAS, TWAS, and EWAS, as well as ENCODE, EQTL data, a series of gene sets data, as well as molecular networks. And the output of Mergeomics is basically two things. One is the gene sets that are associated with a certain disease of interest, and second is the key drivers, or so-called key regulators, of the gene sets. When developing this tool, the goal or the feature we always keep in mind for that we want this tool to have is first, we try to accommodate a multidimensional data and try to be as comprehensive as possible in data integration. And second, we want the performance to be as robust as possible and preferably better than existing tools. And third, we want the tool to be easy and flexible to use so that even biologists not familiar with bioinformatics technology can easily apply the tool to their study. So now I'm going to introduce the several analytical components of this tool, starting from the first component called the marker setting enrichment analysis, or MSEA. What MSEA do is that it takes association data as input, including GWAS or TWAS or EWAS, and then we have a marker dependency filtering module to kind of prune away the dependency between markers, such like linkage disequilibrium. After that, we integrate two levels of data. One is a gene sets data that could be either canonical pathways or co-expression modules, or simply signatures from gene expression profiling. And second layer is a functional genomics data, which could either be ENCODE data or tissue-specific EQTLs data. And eventually, we adopted, in MSEA, we adopted a newly developed algorithm to help us find gene sets associated with disease. Based on simulation, so we test the performance of MSEA across 3 different independent GWAS data sets against 2 other commonly used tools, Magenta and IGSEA for GWAS. And it shows that MSEA has the red line here, has consistently better performance compared to other two tools across different GWAS datasets. Another important component that we have on Mergeomics is called the module-level meta-analysis, or meta-MSEA. What it basically do is to perform module-level meta-analysis across different association datasets. There are two advantages of doing this. First is allow us to kind of parallelly consider independent studies across population, platforms, data types, or even species. And it can also help us overcome some of the technical difficulties that we usually encounter when we perform meta-analysis at the marker association level, such like a meta-analysis of multi-ethnic GWAS data. To prove that this concept will be useful in actual cases, we have a case study that, that was done on the lipid level. So we captured a series of kind of quite plausible pathways related to lipid metabolism. So the things I want to lead you to kind of focus on here is that using meta-MSEA, all these pathways have a quite strong signal strength compared to individual MSEA results thanks to the module-level meta-analysis. More importantly, when compare this result to the traditional, to the result from traditional meta-analysis, we see that the signal strength is significantly boosted consistently across different pathways. In the second case study, we perform kind of do the similar things on the glucose, on the regulation of glucose level. So one thing I want to emphasize here is that in this case study, we perform meta-analysis on 2 different species, human and mouse, and 3 different association data sets, GWAS, TWAS, and EWAS. And this leads to the discovery of, again, a series of quite biologically plausible pathways related to glucose homeostasis. I want to highlight a few pathways here. First is the type 2 diabetes pathway, which is basically impossible to find if you only consider single association data sets. However, in meta-analysis, we're able to capture this pathway. A second pathway is the lipid transport pathway. Again, this pathway only show up for TWAS and EWAS data sets, but was completely missed when we only use the GWAS data sets. But again, in meta-MSEA, we're able to capture this pathway. The next important component of Mergeomics pipeline is called the weighted key driver analysis, or WKDA. What WKDA do is it basically takes the genes in a disease-associated subnetwork, which could be a gene set that's captured in MSEA or any different gene sets that capture from independent sources. And we try to identify the key drivers for this gene set or this group of genes. What a key driver gene means is that its surrounding or its neighboring gene in a regulatory network is significantly enriched for disease-associated genes, for the red genes here. What makes our WKDA different from existing key driver analysis tools is that we are able to consider edge weighting information, which is previously not achieved by any other existing tools. The advantage of incorporating edge weight is that it makes the prediction of key drivers more robust, as can be seen by the simulation result here. So the green bar here is the performance of WKDA, while the white bar here is the unweighted KDA. So we see that WKDA has consistently better performance across different data sets and across different tissues. So the performance is always better than the unweighted version of KDA. Another kind of practical importance of running key driver analysis, especially for biologists, is that it can help us kind of visualize the whole subnetwork, so you can easily interpret the molecular regulatory circuit for individual genes, if there's any genes that you are interested in. So in this way, biologists can easily come up with the detailed experimental plan to kind of verify a specific components of the network centralized by the key regulatory genes. So this is quite practical and useful to design next stage experiments. Okay, so next let's introduce Doug to help to introduce the Mergeomics web servers to us.
All right, thanks, Lua, for introducing the method underlying the web server. So now I'm going to go into our web server implementation. So the reason why we wanted to create a web server is we wanted to make our approach available to a wider research community. A lot of people don't necessarily have the technical know-how to use an R package, and there's a lot of data sets kind of going in and out in this pipeline. So we wanted to develop something that was user-friendly, that would be web-based, and then all the computation would be driven on our end. To the right here, you can see the home page of our web server. Also, another reason why we wanted to do this is we wanted to provide a location for curated resources that individuals could use in their analysis that would potentially be useful. So kind of like an all-in-one stop where it's a lot less difficult for the users to do what they want to do. So here we have a quick overview of what workflow might look like for our web server. Kind of the key takeaway here is that these are the 3 analytical modules that a user would use, and they flow very linearly within each module. So the particular user, depending on what study design they want, they may not want to use all 3 of these modules here, but our pipeline is very flexible and allows the user to use whatever module best pertains to their particular data. We also include a very detailed tutorial and step-by-step walkthroughs of each of the different modules along with example data, so users will know what kind of format they'll need to have their data in order to use our web server. So to kind of quickly walk you through what it'd be like to go through one of these modules. So if, for instance, as Leah mentioned earlier, you wanted to look at marker set enrichment analysis, you would choose this module here, and then kind of walking stepwise through. The first thing you would want is some sort of association data. So if you're looking at GWAS, you would want your particular markers and then your association value with the trait. We look at that at negative log 10 value, or you could select one of our pre-curated data sets. From there, you'd kind of walk through this whole process, kind of uploading or selecting from our curated data sets until you get to the parameters section. So we wanted this to be as faithful an implementation of our R package as possible. So for kind of the power users out there, we allow them to change all of these options that could make a difference in your analysis. However, for most uses, the default settings would be fine for most analyses. So if someone doesn't necessarily have the technical know-how, they could just go with the default settings. From there, the computation will occur on our end, and then the user will be given results, in this case in the form of a ranked data table of the different gene sets which were enriched for the disease. Once they've received their results, if that's all that the user wanted to do, they can stop at that point, or they can choose to continue on to the next module in sequence. So again, we wanted this to be a very flexible and modular pipeline. As far as curated resources, we have a ton of association data, different types of marker mapping data from sources like eQTLs, distance-based mapping, and ENCODE. We have gene sets from canonical pathways like KEGG, Reactome, and BioCarta. Users can also use data-driven gene sets like co-expression modules. And then we also provide a number of tissue-specific Bayesian networks and protein-protein interaction networks. We also allow for file and parameter tracking as you step through the different modules, so you know exactly what you've put into your analysis by the time you've got to the end. This is also output with your results, so you know exactly what you've done to achieve those results. So you can have some form of reproducibility. We also include a built-in network visualization module that's fully interactive. However, we also do provide Cytoscape-ready files if users would like to do that on their own end. And then finally, our website is located here for the web server. We do provide optional email notifications when your job has been submitted, when your job completes. We also do have a Google group for any kind of frequently asked questions or any sort of discussion forum, things like that. And then we also have an R package on Bioconductor. So with that, we'd like to thank our lab, our collaborators, and then individuals at UCLA IDRE, where our web server is hosted. Thanks.